Okay, so today we are going to do our dissection of the lungs, and we're going to be looking just at the structure, but also considering some more questions about the adaptations of the alveoli and the lungs for their function. So these lungs come from sheep. They're usually just considered waste. They might end up being ground down and end up going into probably low quality hamburgers. So here we have our lungs. It's quite a good set of lungs here. We also have attached to here, in between the lungs, a heart. And if I flip this over this way, you can see that would be the front side. Okay, so your heart is there, okay, right in front of the lungs. The lungs are either side. Over here, we've got our trachea, which is the hard. We've got the trachea, which is the hard material coming up here. This one's collapsed slightly, and I'll get some people up to come and feel this because you can feel this hard cartilaginous structure, which is what keeps it open when you're breathing in. And then here you've got your voice box, and here, does anyone want to make a guess what this is? This is the tongue, because your tongue's attached to your back of your voice box. So this is the voice box. And down here we've got the trachea, and then we get down. I'll remove the heart in a minute, and we go down into our two lungs. If this works really well, then we're able to get the lungs to inflate in a minute. We've also got down here at the bottom this thin sheet of muscle, which just about you see, which is the diaphragm. So this is the thing that contracts and goes flat, which is attached to the bottom of the rib cage to allow you to breathe in. So we're going to start now. You're going to do your full heart dissection later, so I'm not going to concentrate on the heart. I don't think this heart's in particularly good condition. It's been hacked around a bit anyway. But I'm going to start just by removing the heart. It's much easier to use scissors for these things. But you can see it's where it's connected. This explains as well why you have a heart and lung transplant at the same time, because they are intricately connected. It's quite tough tissue, this, and blunt scissors doesn't help. So that's our heart. We'll put that to one side. You can see it's not had very good days. It's been kind of cut open on the ventricle that side. Have you done your heart dissection yet? You can do that. Oh, you're going to probably do it next week. And you've got your veins and your arteries there. But I'll put that to one side. We won't be doing that today. Let's start off though by having a look at the trachea. Okay, there's some gloves there. Do you want to grab one? Okay. So I'm going to cut through two things here. The esophagus, which is behind the trachea. It's the soft muscular tube. And we've got the trachea. I'm really going to get a better pair of scissors. There we go. I want to be careful of that because see, it's actually attached to your esophagus is behind your trachea and attached to it. And I'm going to cut away that, and I'm going to cut a bit to trachea just to give it to somebody to have a feel of what it feels like. Here we go. So have a feel of that. Describe what it feels like.
It's like it's tough.
Yeah. Can you feel the structures? You've got little rings.
Yeah.
It's got little rings in it. And so those little rings here, this of course is a tube, it's just collapsed down. I can make it into a tube just by going it through. There we go. And you can feel those hard things. You can actually feel them in your own throat. Just put your hand under your Adam's apple. You can actually feel the cartilage rings there yourself. We're not going to go into the voice box anymore. So how's the oxygen, or the air I should say, go through? It goes through the mouth, down through the trachea, and then it reaches a point here where the lungs split, well the airway splits into two. And that's about here. And this is leading to the two, what are these called? These two airways, one on each side. Bronchi, excellent, bronchi. So you've got 2 bronchi, and then we've got the bronchioles. I mean, I think we're going to be lucky here because these look like they're in pretty good nick. We might be able to get some inflation out of these. Yeah. Trouble is, when they're slaughtered, the animals and butchered, they don't tend to take too much care of these, so you have to try and are especially nice to actually get ones that are worth it. So what we're going to do, we're going to need some help here, is we're going to take a rubber tube. This is going to take a bit of engineering. Let's see if we can get this to work. Okay, and I'm going to try and insert the rubber tube into here. And see if I can get a seal. There we go. That's actually quite good. Right. Right. Okay. Now, need someone brave. Okay. Yes. Can we come up here? Okay. Now, we need a. On my desk, there are some tissues just around there. Okay.
No.
Yeah, just on my desk in there are some tissues. Or I see some paper towels here. Grab the paper towels here. That's it. Right, can you take the ethanol and just put it over there? Right, so what we're going to do, if you go around that side, actually you can stay here, that's fine. We're going to dip this in the ethanol, and then we're just going to wipe it off, okay? So we're going to dip that in the ethanol, wipe it off, so we're sterilising the end. Now, here's a very important fact for you. When we do this, you're going to exhale, don't inhale, exhale into the tube. If you stop exhaling, do not leave it, okay? Because if it then goes down, it'll then push stuff like blood back into your body.
I'm not sure volunteers now.
Tough. So you volunteered now.
So just
There's no going back.
don't inhale.
So basically, exhale and then move it away from your mouth, and if possible, just pinch it after you've exhaled. Okay. All right. So we'll see if it's well. I'm going to pinch to hold this to keep the seal, and hopefully we should see some inflation. Okay. Oh, let's see. We've got a puncher. Okay. Try again. Oh, that's going, isn't it? So see if I can stem the other puncher. Hang on. If I pinch off the bronchiole here, try now. Ooh, look at that. There you go. That worked really well, didn't it? Well done. Okay, you all right?
Yeah.
Okay, right. Thank you for that. Brilliant. First time I had one of those work in a while. They are really hard to get to work because they're butchered. So what we're going to do is now going to remove one of the lungs. Now the lungs and the whole thoracic cavity are covered by something called the pleural membrane, and the pleural membrane is what seals everything in. So if that pleural membrane is punctured, then that causes you to have a hole in your thoracic cavity, which means that when you try and inhale, instead of sucking air, well, the pressure goes down inside your rib cage, but instead of sucking air in through your trachea, instead sucks air in through the wound. And so often that's what happens if you get a puncture wound. Your lung collapses because the pressure isn't maintained, so it starts sucking air in through the wound instead of through your trachea. So this is quite a interesting tissue. It's very soft, it's squidgy because it's mainly air. Right? If you want to bring your hand over, okay, I'll stand here. Just there you go. Just give it a good squish. There you go.
Ooh.
Very, very squishy. Okay,
Yeah.
it's basically then just mainly air, but it's lots and lots of tiny. You can't see them unless you look under a microscope. Lots of tiny pockets, alveoli. And if I show you the bronchi, okay, let's just get it. There's the bronchi here. All right. So you can see the cartilage there. It's that white cartilage. Okay. And then if I cut the lung in half, then you'll see the spongy structure from the side. So, see these little white tubes here? They're the bronchioles, the bigger tubes, and they get smaller and smaller and smaller until you can't see them anymore. And you see the colour, it's nice, rich red. That's because the amount of capillaries that are flowing in. Okay, you can't see the alveoli, but you can feel their structure because it's squidgy. So what are the three things that the alveoli are adapted for to maximise our gaseous exchange from last lesson? You remember that? Three things, three ways in which they're adapted. Yeah, so maximise surface area. If I could stretch this out, it could basically cover the size of a tennis court, the amount of surface area we have there. We've got a maths activity we're going to do later to show that. What else? You tell me? What else have we got? Why is it so red? What have we got flowing around each alveoli? Capillaries. And what's special about the walls of the capillaries and the walls of the alveoli? They're one cell thick to maximise the rate of by, beginning with D, by diffusion. So we're maximising diffusion because diffusion depends on the length of the pathway. The longer the pathway, the longer it takes to diffuse. The shorter the pathway, the shorter the amount of time it takes to diffuse. What's the other thing that maximises the rate of diffusion? The difference in
concentration.
Difference in concentration in the air outside and the air in the blood. And that's why we have to keep renewing our lungs, okay, or breath in our lungs. If you breathe out, you breathe in, you've got 21% oxygen. Goes down to about 19, 18 before you breathe out, okay? So some people mistakenly think all of the oxygen's used. It's not, because otherwise if you gave someone the kiss of life, you'd suffocate them. In the kiss of life, you breathe in and you breathe out air into their lungs, but in actual fact, you're only using about 1 or 2% of the oxygen each time you breathe in. Right, we need the oxygen levels to be quite high to keep doing that. So that's the major structures, and of course then also we've got the blood flowing slowly through our lungs, making sure it's got plenty of time for gaseous exchange. Also, if the blood went through at too high a pressure, it would burst all of the capillaries. And actually that's got a name. Does anyone know what happens when you burst a capillary or a blood vessel in your lungs? It's called a pulmonary aneurysm. It's not a nice thing. Okay, it basically means you bleed to death inside your lungs. So pulmonary aneurysm, unless it's treated really, really quickly, can result in death quite quickly, and it's quite hard to treat. And people who have been smoking severely or have damaged lungs, or things like tuberculosis, which damages the lungs, could suffer from pulmonary aneurysms. Okay, that's the end of our dissection, I think. Just to show you again there, we've got the diaphragm down here, and now this one we don't have. You could also get where you get the liver attached down here, because the liver is just under the diaphragm. Okay, but that's the end, and we'll pack that away. So we'll just have to wash everything down with alcohol to make sure it's sterile underneath. And if anybody wants to come over and grab a glove, not two because we've got quite a few, grab a glove, you can have a hands-on experience to feel what the different tissues feel like. Well done. Okay.